Delivering gene therapy to patients is what UniQure is all about at this point in time. We're publicly listed and refer to our documents that are publicly available for forward-looking statements. We believe that gene-based medicines are going to change the world of medicine going forward, and UniQure is clearly one of the pioneers in this area, which is shown by the fact that we've launched the first gene therapy product in the world in Europe, Glybera. We have recently shown robust Sanfilippo B data from a phase 1/2 study, and at the end of this year we will show data from our Hemophilia B trial. Also, very strong endorsement of our technology and our product assets is the partnership with BMS in the cardiovascular space, which is worth over $2 billion in milestones only. This is a very important partnership for us, and will certainly give us stability to do what we foremost want to do, which is to build the industry-leading gene therapy pipeline. We will focus our own internal efforts on rare and other genetic diseases, and will partner those that are going for larger indications. And where oftentimes gene therapy is associated with genetic diseases for small applications, we think it is actually going way beyond. In our portfolio, you can see drugs like Parkinson's and congestive heart failure, which show the direction that we're going here. So UniQure spent a long time developing a technology platform, and we would claim it is a fully integrated platform today with strong know-how and partnerships on vectors and promoters, which are essential for the quality of your gene therapy. We have a manufacturing site with 50 people built in Lexington outside of Boston and in Massachusetts in the US. We have strong development and regulatory activity. Key thing from a technology platform company like us to convert to one that actually builds a portfolio is to start focusing on the patient. And we're doing this by having reorganized the company from a functional setup in a matrix structure to look at, in particular, the driving elements of our portfolio, which are liver, metabolic diseases, which are CNS diseases, and cardiovascular diseases partnered with BMS. And this is not just an organizational question. We have also hired 3 very strong medics, Dea Corso, a pediatrician and geneticist, Charles Richard, a veteran in the field of CNS with a background in Shire and Wyeth, and then Patrick Most, who is running our cardiovascular business, an academician from the University of Heidelberg. Now, together they are running a already substantial pipeline. The liver metabolic disease area is covering Glybera at the launch in conjunction with Chiesi, our efforts in the US to get the drug approved. We have a Hemophilia B program, which is very exciting. It is using the same gene cassette that was used by St. Jude's in stellar results that you've seen, taking people completely off prophylactic for a long period of time. We're using the identical thing, and you will talk more about this in my talk here. Converting in a modular way what we've learned in Hemophilia B to Hemophilia A, and we are working on a number of undisclosed preclinical programs in the liver space. We have 2 more clinical programs right now in the CNS. One is Parkinson's disease, 4 dose groups, a dose escalation study funded by the NIH and run together with UCSF. This has gone through the first dose cohorts. It is in the second and making nice progress. Sanfilippo B, we've actually read out 1-year follow-up data from that trial only a couple of weeks ago. We're working on a microRNA approach for Huntington's disease and again, multiple preclinical programs that will funnel into our pipeline. And the last area here is cardiovascular, partnered with BMS. CS100A1, a master protein of heart function, was the core thing that attracted BMS in conjunction with our ability to actually manufacture for phase 3 and for market launch out of our facility in Boston. So the fact that we know how to manufacture this type of product for mass applications was critically important for BMS. So looking at hemophilia, I believe that everyone knows what it is. It's a rare genetic disease. Spontaneous bleeds are the key issue, and it is fairly devastating. Today, the standard of care is enzyme replacement, and we think that with gene therapy, you can get away with enzyme replacement with a one-shot treatment. We're piggybacking, if you want, the AAV8 data from a phase 1/2 trial conducted by St. Jude's. And you can see a dose escalation here, starting at a low dose, mid dose, and high dose. And it's important to understand that at the high dose, in 6 patients, there was an elimination of 96% of enzyme replacement that was needed in these patients before. So they're almost entirely of prophylactic treatment. And what I'm saying is if you achieve more than 5% expression of normal, of factor 9 in the body of a patient, you do not need any prophylactic treatment anymore. And you certainly have a very valuable commercial proposition to patients in a commercial environment. Now, we licensed this gene cassette a long time ago. We do not have the rights to AAV8, and we think that AAV5 is actually a better vector. So what we're doing is we're running a new trial using exactly the same gene cassette. We're packaging it into AAV5, and it's manufactured in insect cells. So the question is, does that actually work? Well, AAV5 insect cell-based manufacture works in San Filippo, as we've shown, and you will see in one of the later slides. But interesting here is that we could actually convince both the European Medicines Agency and the FDA to allow us to cross-reference data from the St. Jude's trial and from another trial where we used AAV5 in the context of acute intermittent porphyria, use that safety data and have a very aggressive protocol which starts not at a subtherapeutic dose but at a therapeutic dose higher actually than the one that St. Jude's ended with and showed very clear clinical benefit. And the second dose we're using is 10-fold higher than the St. Jude's highest dose. So we're expecting to get higher expression than was seen in St. Jude's, and we're very much looking forward to the data from this trial. But I would like to say also that this fact of being able to cross-reference in gene therapy using the same elements again and again in similar diseases where you're just exchanging a gene can be very powerful to build a strong pipeline fast. So the population is 10 patients. The first 5 are recruited and are currently being dosed. We're expecting to show data from these patients regarding safety and tolerability and also expression levels in those patients by the end of this year. Maybe not all 5, but we're on good track for this first cohort of that trial. So very excited about that, and we think that where we are today with our expertise in being able to manufacture it to a market introduction, we believe that we are actually going to be first in the market. San Filippo, again, a devastating disease. It eliminates the kids' capability to progress and develop skills at the age of 2, and at the age of 4, typically they fall off a cliff and completely lose their intellectual capabilities. And they are lacking an enzyme called NAGLU, which is important for emptying the neurons of metabolic residues. And if you can't do this, it's like you're carrying stuff into your kitchen all the time, but you're never taking anything out. It's just building up clogging masses of metabolic residue, which is in the end destroying these neurons. So it's a significant medical need. And with our partner, Institut Pasteur, we've run a trial on 4 kids and are showing 1-year follow-up data. What is remarkable, first, is that safety and tolerability in the brain was established. But even more important, we can measure in the CSF, and this is actually delivered into the cortex and cerebellum through burr holes. We can measure in the CSF, which is a spillover effect, 14 to 17% NAGLU enzyme activity, and think that is clinically meaningful because they're attenuated forms of San Filippo where 3% actually makes already a massive difference on quality of life of these patients and also allows them to survive much longer. So this effect is persistent and at the same level, 14 to 17% over a full 12 months. There's continuous incremental cognitive development across the 4 patients, which again is very strong. The oldest was almost 4 when he was dosed, and they continued to develop skills over that 1 year. So we're very excited and as a company have committed to taking this program into pivotal trial. We come to the last program that I would like to touch upon. It's not yet clinical, it's preclinical. We have certainly learned from other programs in the industry by optimizing the gene that we're using, which is a master regulator and a top-level hierarchy regulator of cardiac performance. We have used not a standard promoter, but a heart-specific promoter, which also allows us to basically have it act as a switch so that it doesn't create any side effects anywhere else. We have a heart AAV that delivers very nicely, and we're delivering actually retrograde in a bolus where we expose the heart for about 30 to 40 seconds to the gene therapy solution that is being pushed into the heart muscle. And so far, we have had extraordinary results in the context of pig studies that we've run. Here is a comparison of sham, and each group has 20 pigs. The second one, the red line, is actually the luciferase saline, where you can see that you have a survival rate in a human-relevant pig model of 10% using saline and luciferase in a gene therapy application after a provoked myocardial infarction. You have 90% survival in using S100A1, which is a complete reversal of the disease and something that hasn't been seen before. 6 months biomarker troponin T and BNP are showing similar results. So again, we're very proud of this, and we have to push it forward now, get it translated into humans, and show that we can achieve a similar proof of concept in congestive heart failure in patients. And obviously, this is a market of 20 million patients today, growing rapidly with the aging population. So this is where we take gene therapy from monogenic diseases, small populations, into the field of non-monogenic and larger populations. Obviously, this data was what attracted BMS to close this deal with us. I would like to highlight a few things. They thought this is their entry gate into gene therapy, so S100A1 per se was massively interesting to them, but they wanted to run another 9 programs that were starting one after the other with BMS. They invested up to today 9.9% in UniQure's equity, and they gave us a significant down payment. So at this point in time, we're working on 4 programs. We have received $140 million upfront, which are completely unrelated to the program, so we can use that money to build our own pipeline because BMS is funding the entire research and development. In addition to that, they will be paying milestones to us, which could, at the maximum, equate to over $2.3 billion. So this is a very strong partnership, and it gives us a very strong backbone, if you want, for our cardiovascular disease to focus our own efforts to the CNS and to the liver space. Coming to my last slide, 2015 clearly was and still is a breakthrough year for the company. The Sanfilippo B human trial showed for the first time proof of concept with a gene therapy in a lysosomal storage disease. And with the modularity of this concept, we think we can take that to other lysosomal storage diseases as well. The BMS partnership I just mentioned, this year Glybera was launched and pricing established in one country in Europe, which is Germany. We have established the 3 indication areas, which we think are very powerful now in helping us drive patient-oriented drugs to actually the market. We have raised $220 million to date through the BMS transaction and a follow-on at NASDAQ. And at the end of this year, we will be showing human proof of concept preliminary data from our Hemophilia B trial. So the company is on very good track. And I can only say I'm very pleased that we stayed persistent with Glybera and got to the point where gene therapy gets accepted in our industry as a viable modality that will change and revolutionize the way that medicine is being done, as it is curative through single treatments. Thank you very much.